Hi everyone, this is Dalwin Jacoby, and welcome to this little mini lecture on chromosomes, genes, gene map locus, and the naming of particular genes. This is a portion of NURS 806, Genetics and Genomics and Population Health, for the Spalding University Postmaster's DNP program. We'll start off and do a little quick review. I know you have already gone over much of this material in your web-based mini course, so we'll do a little quick review as an intro to the gene map locus and the naming of genes. We'll start off again and mention chromosomes, and we all know that these are found in the nucleus. The chromosomes themselves contain genes. The chromosomes occur in pairs. In the normal human cell, all cells within the human body supposedly contain 46 chromosomes. We also know that different species have different numbers of chromosomes. And I'll also point to the pictures here, the little schematic here on the right that shows a particular chromosome in a certain phase of mitosis. And at this particular phase of mitosis, you see that the chromosome has two arms. The short arm, remember, is called the P arm, and the long arm is called the Q arm. And the little pinch in the middle is called the centromere. This is very important when we start talking about naming genes and their locus or their location. Let's talk about genes. And realize here that genes are located within the chromosomes. The genes are the basic units of inheritance, and as we know from your previous coursework earlier in this course, they are composed of sequences of DNA. They code for the proteins in the body, and thus they influence all the aspects of human structure and function and structure within the body. So realize that in this schematic here we have a cell, we have the nucleus. The chromosomes are found within the nucleus. The chromosomes have a short arm called the P arm and a long arm called the Q arm. The chromosomes, as you unravel the chromosomes, you will have those sequences of DNA, and certain sequences of DNA are called genes. Now it's really important to note that those sequences of DNA, not all of those sequences code. And this is really important when we get down the road and we start talking about genetic testing, we talk about exome testing. You need to know the difference between an exon and an intron. So realize in a gene, within a gene, a particular gene, or a sequence of DNA, that certain sequences code and certain do not. Those that actually code are called exons. And you see in this little schematic here, it's kind of giving you an example of an exon versus an intron. And remember, most of the genetic material, most of the DNA does not code. So the majority are introns, but those that actually code for a protein or a structure within the body are called exons. DNA transcribed to messenger RNA consists of coding sequences of exons only. The intron itself, which is shown in the picture here, is a non-coding sequence of the DNA. And actually, the introns are spliced out of messenger RNA prior to translation. So next, we'll look at those particular chromosomes, and when they're lined up in that particular sequence in metaphase, and they have the P and the Q arm, and they have the centromere, the little pinch within the middle. And scientific research has shown that we can actually stain these chromosomes, and when you stain them, they produce sequences of dark and light cross bands of varying length, and they are particular to certain chromosomes. And interestingly enough, we can use them to actually map out the specific location of particular genes. So when we talk about a locus, that is the location of a particular gene on a particular chromosome. So when you talk about that locus, you've got your exact site on a banded chromosome. So to identify this locus, you're going to say the number of chromosomes, like 1 through 26, the arm, whether it's the P or the Q arm, which is either short or the long arm, the region of the arm in the specific band. So this is a really interesting way and a really neat way to actually look at the exact location of particular genes. So let's look at an example here. Let's look at the gene locus and the particular name of a particular gene. So let's start off with the CTFR gene. It has been named because it is specific for cystic fibrosis, And there is a change on the gene in this particular locus for cystic fibrosis. So the 7q31.2 location is the exact locus for the CTFR gene, which means it is on the 7th chromosome, on the Q arm, at the 31.2 region and specific band of that particular gene, that particular chromosome. Let's look at another example. Let's look for the BRCA gene. You've heard of the BRCA gene. BRCA1 is a gene. We know there is changes in the particular gene related to breast cancer. And we know that it is located at 17q21, which tells us that this particular gene named BRCA, breast cancer gene 1, is located on the 17th human chromosome on the Q arm, which is the long arm, at the 2-1 region of that particular arm. So thus ends our little discussion on chromosomes, genes, gene map locus, gene banding, and gene naming. Feel free to view this as often as you want. It's got some pretty important information, and we'll work to add more onto this information later.